So hello everyone. I'm going to talk to you, as Emma said, about the microbiome build-up on domestic washing machines and linen. And realistically, when we're talking about this, the conversation started around healthcare uniforms being laundered at home. And as this journey over the last 10 years of looking at textiles and as fomites has come out, there's a lot of other scenarios where domestic laundering is used within healthcare facilities. So I know neonatal wards use domestic machines, care homes, residential mental health homes also use them. So actually, this is a much broader area than most probably healthcare uniforms being laundered at home. So I'm going to show you some data on the survival of bacteria on textiles. We're also going to look at the actual efficacy of using a domestic machine in decontamination, and also the contribution of antibiotic resistance within these environments, and also how dirty is our bed linen and how quickly does it become dirty, which is rather gruesome when we start looking at the data. I have to say that the men come out a lot worse than the women. Not sure if that was what we'd be expecting. So as I mentioned, in the UK and the USA, domestic laundry machines are commonly used for healthcare laundering. And really the concern around decontamination and the spread of AMR in those environments is something that we do need to think about. So many years ago, the starting point was, let's just see how long the bacteria survives on different textile types. Let's just go back to basics here. So we chose the Staph aureus, which is a skin microorganism, and then E. coli, which is a gut microorganism. So we had 1 gram negative and 1 gram positive, and we thought most textiles used within the healthcare environment are either the cotton, polyester, or polycotton. So let's look at the survival on these different fibre types. And as you can see from this, that on the cotton, both microorganisms are surviving for up to, I think we went up to 21 days here and decided that was long enough. Now, part of the reason for this occurring is because you've got nutrients in the cotton, and there's some moisture content. So it is quite a good environment for the microbes to survive. On the polyester, it's a much shorter period of time, so we're looking at 200 hours there, which I think about 7 days, and then you're seeing a dramatic drop-off. But what we're actually seeing is the microorganisms are just sitting on the surface of the polyester. So the way in which they behave on the different fibre types is significant to how long they survive. So when we're talking about, well, what's best cotton, polycotton, there's lots of pros and cons. And what's interesting is during the pandemic, we did work on coronavirus, and we were actually seeing the opposite. So we were seeing desiccation of the coronavirus in the cotton where it was absorbed, and what we were seeing was the virus was just surviving on the surface of the polycotton fibre and was more easily transferred. So just this is because how bacteria are behaving. It doesn't mean that's exactly how the virus is going to behave. And we have done some work actually on Candida aureus, and that survives relatively well on the surface as well. So not just my word for it, we've got some nice images here. So in the polyesters, you can very much see that the microorganisms are just sitting there happily. Whereas when we start looking at the cotton, we're seeing much more clumping and building up of potentially, which could over time become more of a biofilm type structure. I'm not saying it is a biofilm, but you're starting to see those communities, and we've got much longer survival on that. So we know that microorganisms are surviving. We have a PhD student in the lab at the moment that's looking at cross-transfer from cotton and polyester as well, and we're seeing that it's still transferring up to a week, so that's hot off the press data. And so there is a risk there of transfer. So what's happening during laundry? So it's there, it can transfer to the environment, but are we effective at then removing it in the domestic sense of a laundry machine? And again, what we did was we looked at the same two microorganisms, but this time we did look at a polycotton and a polyester contaminated textile. And we looked at it after survival at 40 degrees because the NHS policy says wash at 60 with detergent and you'll remove microorganisms. And that is absolutely right. If you wash at 60 degrees and your washing machine reaches 60 degrees and you use detergents, you will get rid of these. But I would honestly like to ask those in the room, how many of you go home and wash items of clothing at 60 degrees separately on a reasonably long wash? And I can almost assure you, I can see head shaking. No, we've all got busy lives, we're all saving the environment, etc., etc. So most of us are using 40 degrees and more rapid washes, which I will come back to. So when we looked at just a normal 40-degree wash, and we've got survival of the E. coli after laundering at 40 on the contaminated and the sterile fabrics, so which you can see here. So these are sterile ones. What we were basically seeing was that we were just transferring things around the washing machine. Okay, so we started with 10 to the 8. We were left with 4.5 logs on the washing machine, but the other bacteria had just gone across to sterile items in the wash. So it's just moving it all around a little bit in the domestic machine at 40. So we don't need to just take my word for this. There's been lots of studies that well documented the survival of textiles, and we've got E. coli, Staph aureus, Enterococcus, Candida, SARS, HSV. So it's well documented that a whole range of microorganisms can survive on textiles, yet they're most probably not given, when we think about domestic laundering, the same priority as a hard surface when we're thinking about the hospital environment. And there's also a whole range of literature that shows that these microorganisms are able to transfer to surfaces around us. So this has been shown on textile skin, stainless steel, flooring, surgical gowns and more. So there is a growing body of evidence, and has been for a number of years, on the fact that textiles are fomites. So that's been well established. But then what we decided to look at was the domestic machines and their actual performance under a number of different conditions. So the poor microbiology team all got roped into this experiment. So this is basically the microbiology team's washing machines that we're going to look at in the next few moments. And what we wanted to establish via a test method that I'd developed for the global industrial laundry industry is a bioindicator method using Enterococcus, which is thermally tolerant. So we're looking at a worst case scenario within a domestic setting. So these bioindicators will, within the next few months, be used quite broadly across the laundry industry. But what can they then do in comparison to tell us about what's happening in the domestic environment? So within the bioindicators, you've got a number of different logs going down from 1 million cells to 3 logs. And we were going to wash it at 60 degrees, a full-length program, one of the rapid length programs, which is a lifesaver in our house with children around and needing sports kits and all sorts. And then we looked at the biological and non-biological detergents. What's really happening in our laundry machines. So as you can see, the microbiology team had a whole range of different machines, which was really quite useful. Different models, and also a range of different ages of washing machines, which is going to become quite an important point in a moment. So what we did was we put in the bioindicators, but we also put in an iButton temperature monitor. So we monitored the temperature continuously through the cycles. So we were able to see, is it killing the microorganism? And is it reaching the 60 degrees that we actually put the machine on? So as we can see here, we had 3 machines that didn't quite get to 60, but maybe if we have a 2-degree standard deviation, we could sort of say, well, they did reach the 60. And the 60 degrees needs to be reached for a minimum of 10 minutes to decontaminate. So we were looking at the time as much as the temperature. And so we had 3 machines out of 6, that's 50%, and yes, it is a small sample, but we were looking and seeing, I actually think this may get worse if we did a bigger sample rather than better. So we had 3 machines that we were happy with. This machine here was at 20 degrees. It never left 20 degrees ever. And my colleague says I still owe him a washing machine because he never knew that he was just washing his laundry in cold water. But he's still living, so we're all right. But I do owe him a washing machine still. And what we can see then is the other 3 machines are either way off temperature or way off time. And this is meant to be in a full length cycle at 60 degrees. So part of what I'm saying is you can be taking your laundry home, you think you're following the guidance, but no one's monitoring it. How do we know? So that in itself is scary. So our machines are not reaching the temperature and times that they should be. We then did the rapid cycle to see, so this again at 60 degrees to have a look at what was happening. And this time we only had 2 machines that actually passed the 60 at 10. So we're progressively getting worse, and the 4 that didn't. So you think you're washing it properly on the rapid cycle again, and you're absolutely not. And the performance is not as good when we're using rapid cycles, which to a certain extent we'd expect, wouldn't we? Because it's not in the washing machine for very long. So what does this mean in terms of decontamination with those bioindicators that were in the machine? And basically here we've got all the different conditions, and red are fail and green are pass. So depending on the make, model, age of your washing machine, it can really be hit and miss as to whether you're actually decontaminating what is in your machine anyway. There was definitely a correlation between machines that were over 4 years old and their performance in what we saw in the sample we did. So really what we're saying here is if you want to decontaminate textiles, ideally you'd be getting a new machine every 4 years, which I don't know how good that is for the environmental status really, is it? So it's not great, but that would be my recommendation. And what we then did was said, okay then, so our washing machines are not performing as we think, and 50% at least of us in this room are not washing at the temperature nor decontaminating our laundry. What's actually in our machines? How much are they contributing to potentially cross-contamination and antibiotic resistance? So what we did was we took the rubber of the washing machine, samples from the rubber of the washing machine, and also sort of behind the drawer where the pipes are as well. And what we do need to consider is obviously the water going into the machine will have some contribution to this from the environment as well. So we do need to consider that. And then we sent DNA off, we had it shotgun, metagenomics done of this. I will say, I disinfect my machine monthly, and there wasn't enough genetic material on my machine to actually carry this experiment out. So, and I hadn't done it just because I knew that they were going to swab my machine. So what I would say is a good disinfection for your washing machine does prevent build-up. They really do what they say on the box. So we had a look, and this is basically what we found in the washing machines. So we've got the bacteria genus here, and we've got the class here, so they're just broken down slightly differently. And as you can see, there was a lot of Pseudomonas type microorganisms in there. You'd sort of expect that. We had the Mycobacteriums as well, but there was a real mixture, and there didn't seem to be a pattern. We thought maybe working in the lab, we'd see some of the microorganisms we were using in the lab. We thought there might have been a general pattern, and there wasn't. So it really is very much on your home environment, you as a person, your microbiome, the water, everything together. We couldn't really distinguish that. But what was quite scary was these were all the antibiotic-resistant genes that we found within the washing machine. And if you look at most of these, they were efflux-based. So this is where the bacteria is pumping the antibiotic out. And we were also seeing ones that were very much linked with disinfectant agents, which again you'd expect because you're putting that detergent into the washing machine. So you'd expect to see these types of things. But there was a definite abundance of antibiotic genes in there. So if we think about healthcare uniforms coming into the washing machine, they're going to have some antibiotic resistance. We're then getting the microbiome building up within the machines. I've also shown you evidence of cross-contamination within the wash, because that's the other thing the guidance states, is that the uniform should be washed independently on their own without any other washing in there. And I'd like to know what busy nurse has time for that and enough uniforms to be washing their uniform on its own and between every shift. So we were seeing, which I thought was rather worrying, and again, we had an abundance of different genes across different areas so from the pipe and from the rubber and across all the machines. They were well distributed across them. So we weren't seeing one particular machine was significantly worse than other. It did commonly seem to be building up across all machines, which is worrying. It is slightly worrying. So, okay then, so what's the next thing? We're seeing it there, does it matter? And we were seeing the efflux pumps, and we were seeing the resistance to the detergents as well. So what role is the detergents that are going into the machine? So I'm going to ask a question of you all. How many of you know the volume of water that goes into your machine, the amount, concentration of detergent that meant to go into that volume of water in order to be able to get the correct concentration within the wash? Are you just like me where you go, oh, that'll do? Yeah, it comes out smelling okay, it's clean. Okay, so we're asking people to wash healthcare contaminated textiles in a washing machine that we don't know whether it's reaching temperature, we don't know whether it's reaching time, and we're asking them to use detergents that we don't understand how to dose into the machine to get the right concentration. So what does that mean? Well, what we did was we took the detergent and we looked for a sublethal dose. So we looked for a dose. We did use non-biological because biological's got enzymes in it, and that was going to mix the whole process of the investigation up because there'd be other things going on. So we went with the non-biological. And what we said was, let's find a sublethal dose so we're only killing 80% of the microorganisms that are there, and then we're going to train our microorganisms to become resistant to the detergent. So every time you're dosing in your detergent and it's under the dosage that we should be, not if it's over, it's not too bad, but if it's under, what would be happening within your washing machine that's already harbouring a significant amount of bacteria and antibiotic-resistant genes? And so we trained it. So every time we took some of that sublethal dose and subjected it to the detergent, if it survived, we then subjected it to a slightly higher dose, and then we took some of that one that survived and we subjected it to a slightly higher dose, and we did that up to 20 passages. So we just kept very gradually giving it a little bit more detergent each time, and we wanted to understand whether we could make resistant strains by that process. So here is the organisms that we use, Pseudomonas, Klebsiella, and Pseudomonas, and we use both powder and liquid forms because we all use both. And we looked at the starting concentration, the maximum concentration, and how much of an increase the bacteria had tolerance to on the detergents, and some of these are really quite shocking. So in most cases, we could train the bacteria to be resistant to the non-biological, detergent that we use in our washing machine by just slightly underdosing it. And as many of you in the room nodded and admitted, we don't know if we're dosing it correctly anyway. Okay, so that's fine. What does this mean for antibiotic resistance? There is the science of if organism becomes resistant to a disinfectant, that you can have cross-resistance to an antibiotics. So we took our bacteria that had never been subjected in any way to antibiotics and tested if it was antibiotic resistant or not. So we've got here the zone of inhibition before exposure. So the blue dots are where we started. This is for Pseudomonas. The zones of inhibition after the number of passages it had got up to before it died. And what we're seeing here is green areas are susceptible. The white area is like an intermediate, and the resistant areas is the orangey colour. So what we were seeing generally in Pseudomonas, we weren't necessarily seeing a switch from susceptible to resistant against all these microorganisms, but we were definitely seeing a shift to increased tolerance of the antibiotics in Pseudomonas. So then we did the same, which one's this, for Klebsiella. And again, we're seeing a few that are shifting quite largely. So here we have gone from susceptible to resistant, and here we've gone from susceptible to resistance. So in Klebsiella, we were starting to see where these microorganisms were becoming resistant to the antibiotics used in the hospital environment. And then we got to the staph. We basically took and changed an MSSA strain, so a susceptible strain, into a resistant strain without ever having it been near an antibiotic, just by subjecting it to the washing machine detergents. And you can see massive shifts here in so many of the antibiotics. And that's worrying, because we don't understand our volumes within our domestic machines and the amount of detergent that we should be putting into the machine. So where we got to then was isolates that produce clinically relevant resistances are all listed here. So it was mainly in the staph. We've seen a tiny bit in the Klebsiella to the powder. And you can just see the whole list of antibiotics that we were starting to get resistance to. So is it the fact that we're not decontaminating it, or is it the fact that we're making our domestic machines almost a harbour for antibiotic resistance? So we did genome sequencing of the cross-resistance to see what was happening, and unsurprisingly, we found a couple of genes, both of which were linked with efflux pumps. So we were starting to see that the resistance had occurred in the strains because of the efflux pumps that had been switched on by the sublethal doses to the laundry powder, basically, is what we think was happening. So that there's cross-resistance via this mechanism where they were just pumping it out. So in S. aureus, we saw this MGRA gene, and in the Klebsiella, we saw the ACRB gene. So we then did it with industrial laundry detergents, because it's just important to establish what's happening in those machines. And across all the different ones that we tested, we could get SDS, which is a detergent, not a disinfectant, to go up to 7 passages. Peracetic acid would only get up to 3, and then it was dying out. We didn't test the hydrogen peroxide because the laundries were saying they didn't use it. And the chlorine, again, we could only get it up to 2 passages, so we weren't getting enough resistance occurring to really be able to establish whether we were getting resistance or cross-resistance to antibiotics. We did look at the SDS, where we did get the 7 passages, and we did get some resistance to penicillin. But what I would say is in industrial machines, the SDS is never used on its own. It's always used in combination with a disinfectant, which we obviously couldn't achieve what we'd seen in the domestic machines. So there's the paper if anyone's interested, and it's freely available, so you can have a little bit more look at that in depth if you want to. So it's an open access one, so I'll let people take their pictures. I've always off it, that's it. Good job, okay. And we did put some recommendations into that paper, and we broke it down into healthcare workers and the employees. So we were really saying that to ensure you got disinfectant with machines that aren't warming up, you need to go way above temperature. You need to clean the machine regularly, no mixing of laundry, you need a new washing machine every 4 years, you understand the detergent. We were saying recommendations for the healthcare employer was they needed to establish a list of detergents or disinfectants they thought worked. They needed to offer servicing and performance and maintenance on people's machines if you're going to ask them to use it. They could offer on-premises laundries. They could offer outsourcing. They need to source uniforms as well that can actually be washed at 60 degrees, because this is the other problem you sometimes see. And that they needed some compensation for the renewing of machines so regularly. So then what we had a look at, which was a bit of a side project, was what's happening on our bed linen. Because we were doing a lot of interviews with care homes around their laundry policies and practices. We had over 1,000 care homes involved in the study. And one of the things that came out of that, and then a follow-up focus group, was that unless a bed linen is soiled, which obviously has changed the bed linen, that the policies varied, if there was a policy at all. Some was between patients, some was certain days. So it was very, very rarely, it was very varied, and we felt there was a need to understand what was happening on bed sheets, to understand where the science was, to inform when to change a bed. And I thought, well, this is silly, I'll go and look in the literature. But by the end of my literature search, I understood chimpanzees' beds better than I did human beds. So I thought, maybe this isn't silly, maybe we do need to look at this a little bit. So we did a study where we asked, again, my poor team and some others, they all must hate me, their washing machines and their bed linen, especially where I made them sleep in the same bed sheets for 4 weeks. So there was a lot of fun in our lab. So we got everyone to lay on the bed sheets for a number of different time intervals. Then we looked at the pillowcase, the hips and the feet, and this is where the men failed, I have to say. And then we took those samples from all those different areas, and we just plated them up on both fungal and bacterial plates, just to see what was going on more than anything. And then we also kept the DNA as well, so we could see what was there, as well as how it was building up. So colonization was actually happening really quickly. Within the 3 days, we had a well-established microbiome on the bed sheets. We're actually going to go back and do 1 day, because I want to understand if it just appears as soon as you sleep in the bed sheet, or if there is a slight build-up in those days. But we've been waiting, because we did this in the autumn time, so in the next few weeks we're going to do it again, so we've got the same temperatures, because I think they'll be very different if it's a hot summer's day to it would be an autumn day, so we're just holding off a little bit on that. And as you can see, it doesn't get any worse. And me and my postdoc, when we started it, I said, no, it's going to build up and it's going to get worse. She said, I don't think it is, Katie. I think it's going to be a flat line. So she won that bet, because that is what happened. And what we're seeing is both the bacterial colonization and fungal colonizations around 4 logs. Now, that isn't surprising. I've done a lot of work on curtains, bed curtains, and our lab coats in the lab. And whenever I seem to be testing the textiles in different environments, it always comes out around 4 logs. So this isn't unusual, but we were seeing these we were seeing this different build-up, well, about the same build-up. We did look at it under gender conditions, and as you can see, the male feet were just there, nice and high within a few days. Generally, us women did catch up over time, but they were always just a little bit ahead. We were seeing that happen. But there was no, apart from the feet, we were seeing no real gender distinction. The men did colonise a little bit sooner, both with fungal and bacteria, than the women did. But within 7 days, it was much more evened out. So there wasn't much different there. So then as I said, we wanted to understand what was there. And in total, among 81 samples, we found 1,251 different species of bacteria. So there was an awful lot going on. And again, this comes down to your own environment, to your microbiome, etc. Now, these are all healthy individuals. And then we found 51 species that are in at least 80% of the samples. They were much more the baseline of the samples. And as you'd expect, a lot of them were Staphylococcus, skins type 1, Streptococcus, and some other ones you wouldn't necessarily expect. And then fungal identification, again, we had nearly 300 different species of fungi in that area. But there was only 3 species that were universal, so this seemed to vary quite a lot more between individuals. And then you can see there's some different fungal species there, but all species were present from the 3 days again. So it's very much about quickly colonising them. So my question here would be, if you've got a vulnerable patient, or you've got someone with an open wound, or you've got someone with ulcers, etc., is what should that bed policy making be, really? So on the 3 days, we're seeing it a little bit of a rise to 4 days. So we're really recommending at the 4 days, the bed sheets need to be changed because it's fully colonised by that point. But then I think this needs looking at in more depth with vulnerable patients, and also thinking about the policies around bed changing. This is just a small snapshot of what we found, and I'm looking forward to getting the 1-day data so we can see if it is on a trajectory or it's just happening. We will see. So microorganisms are able to survive on textiles. They are able to transfer. We've got data as well. Domestic machines do not work to their thermal settings that we believe they are, and they do harbour antibiotic-resistant bacteria. Non-biological detergent can lead, the use of it can lead to cross-resistance to antibiotics, and we definitely recommend that bed sheets are changed on the 4th day with vulnerable patients as a maximum. When I say vulnerable, I mean more infections of wounds and things, or someone that's maybe immunely suppressed.